Hello, good evening. I'm Kathleen Klaas. I am working in the Center for Medical Genetics at Ghent University Hospital in Belgium. And here I would like to give you a brief introduction about variant classification of the BRCA variants, which are of course playing more and more an important role in breast ovarian cancer. And now more recently, we also get more and more requests for prostate cancer. We will start with a brief introduction about the type of variants we are seeing in these genes, and then my colleagues will explain you in more detail how the variant classification can be done for the specific variants. Yeah. So this is a training video, therefore the aim of this video is to be educational. So as an introduction, I would give you an overview of the variant types that you can find and that need to be assessed for pathogenicity. And afterwards we will continue with the guidelines that have been published. So, as many of you may know, there are different types of variants you can identify in the human genome. The most easy one to understand are the single base substitutions that may either lead to non-synonymous variants. This means that you get an amino acid change, as illustrated in this example, where the last nucleotide is substituted, A by C, and instead of glutamine, there is an aspartic acid. Or you can have synonymous variants that are silent mutations that results in a codon that codes for the same amino acid. So here you see the same sequence where the A is substituted by a G, and in that case the amino acid remains the same. Synonymous variants, the chance that they are pathogenic are much more than for variants that are that lead to another amino acid, because then the characteristics of your protein may change. Substitutions may also lead to nonsense variants. This means that an premature termination codon is introduced. So here you see an example where the first nucleotide of this codon, the CAG codon, is replaced by a T, and a TAG is known as a termination or a stop codon. So especially the variants in BRCA1 and BRCA2 are lost that lead to loss of function, like nonsense variants, cause or can be considered as probably as likely pathogenic. But there are exceptions about which you will hear later. Also very interesting are the variants that are located nearby the coding exons. So because they can lead to aberrant splicing. This may be this may be just substitutions in the non-coding part of the gene. So and they are mostly located at the beginning of an intron or at the end of an intron. Also, at the branch sites, there may occur some substitutions that may lead to aberrant splicing. However, in practice, it will be more difficult to find this kind of variants, as for many exons, we do not exactly know where the branch site is located. But important to keep an eye on the intronic sequences, especially the first two nucleotides of an intron and the last two nucleotides of an intron are obviously very important to allow correct splicing, as deletions, substitutions, insertions in these sequences may lead to aberrant splicing and therefore pathogenic effects. What can be the effect of variants in these locations? Well, it can either be that there is a skip of an exon, which occurs more frequent. Alternatively, you may introduce alternative 3' or alternative 5' splite sites, whereby you extra intronic sequences are included in the final transcript. There may also be some things happening like new splice sites that occur within an exon and therefore a part of the exon may have been skipped. Another effect that may occur is intron retention. This means that a whole intron may be included in the transcript, or a part of an intron may be included as a kind of pseudo exon. Next, there are also deletions or insertions that may occur, and the first type are frame shifts that cause a frame shift in the reading frame, so that cause a shift of the bases, and hereby there is a complete change of the amino acids after this point of the mutation, where the deletion takes place. So you see that a completely different amino acid sequence is generated. If you have such a frame shift variant mutation occurs when there is an insertion of deletion of the number of nucleotides that's not a multiplication of 3. So if you have an insertion of 3, 6, or 9 nucleotides, or deletion of 3, 6, 9 nucleotides, there will not occur a frame shift. It's only in the case it is an insertion of 1, 2, 4, 5 variants. Nucleotides this leads to frame shift variants, and often frame shift variants leads to generally spoken frame shift variants lead to a premature stop codon. More difficult are to detect baby rearrangements. So this means that there may be a large number of nucleotides that may be deleted or inserted, and this kind of variations may not always be detectable by the standard PCR-based techniques, or also with the NGS-based techniques. You may miss large rearrangements, although in some populations they might be recurrent in the BRCA1 and 2 genes, and they may lead to a faulty protein. How do we deal then with all these variants in the diagnostic lab? So many years ago it used to be very simple. We had either mutations or polymorphisms. At the moment, this terminology is not advised anymore, so it's not used anymore. We use the word variants, and we have different classes of variants. We have variants that are clearly pathogenic, and we have variants that are clearly not pathogenic. But in between, there is a kind of gray zone. We have the variants that are likely to be pathogenic and variants that are unlikely to be pathogenic. So this means that they have less than 5% chance to be pathogenic, or they have more than 95% class 4, more than 95% chance of being pathogenic. But you see, between the 5% non-pathogenic and the 95% for sure pathogenic, there is a whole broad zone which are the variants of uncertain significance. So what kind of variants do we expect in the different classes? Well, for the nonsense variants and the rearrangements and the large rearrangements, they are in almost all cases clearly pathogenic. So for the nonsense, we need to be aware that in BRCA2 there are some nonsense variants that are not belonging to this class, but the large majority are just pathogenic. For frameshift and splicing mutations, they are likely pathogenic, and in some cases we will need some further work to evaluate if they are really for more than 95% sure pathogenic. More difficult is the classification of missense variants. For these variants, in many cases, there might be sometimes lack of evidence about their pathogenic effect. This does not mean that there are no pathogenic missense variants in BRCA1 and 2. It's just the message that I want to give, that many of the missense variants we identify may not be easy to classify. And they're not pathogenic and likely not pathogenic, or often variants that do not lead to amino acid changes or that are very frequent in the general population. Why is it important to use these different classes? Well, it is because there are clinical implications about that. So for the likely pathogenic and the pathogenic variants, these are clinically actionable. This means that patients can be need further follow-up clinically, and it may also have therapeutical consequences, and it may also lead to predictive testing of their relatives. For variants that are classified as class 3, these are clinically not actionable. So what we advise there is that surveillance should be based on the family history and the personal history of the patient. So it does not mean that we did not identify something of importance, but that there is, we just cannot use it for clinical implications. Then we have the not pathogenic or the likely not pathogenic variants. These are not clinically actionable, and generally we do not use them as for clinical. Well, we do not mention them in the reports. So to help you to do the classification of the variants, guidelines have been published. And a very good literature can be found in this publication that was published in 2015 in Genetics in Medicine. And this is kind of gold standard that we are now using for interpretation of genetic variants. So for the purpose of this assessment scheme, the BRCA variants have been assessed using these criteria. But in some cases, we had the criteria are not very clear, and they have been modified according to recommendation of the ClinGen SVI group, and this resource can be found through this website. First of all, what's said in these guidelines is that we should use uniform and standard terminology to describe variants that are identified in genes that cause Mendelian disorders. So the class 1 variants are not described as clearly not pathogenic, but are described as benign. Class 2 are likely benign. Class 3 are uncertain clinical significance, and then the class 4 are likely pathogenic, and the class 5 are the pathogenic ones. When what are criteria to provide, well, to state that something is pathogenic or likely pathogenic or benign or likely benign? Well, there are different scores that can be applied, and this is a kind, they correspond to some 2 points. So we have some arguments that are very strong, and this helps you to discount for a lot, to put it in, to classify it in as a pathogenic variant. And these are called as PVS, which means pathogenic very strong one, the criterion 1. In the criterion strong, we have 4 criteria. It's pathogenic strong, criteria 1 to 4. There are criteria that are suggesting pathogenicity, but the weight of it is only moderate, so they are called as PM, abbreviated as PM, and there are 6 of these criteria. And then we have some criteria that are just supporting, and this is abbreviated as PP1 to PP5. Really, concerning the benign categories, we have some criteria that are standalone. So this means benign, a standalone criterion, and this is a criterion that really relates to the frequency in the population. So you can assume that variants that are very frequently identified in a healthy control population can be considered as benign. We also have criteria that are strongly suggestive to be a variant benign, so it's abbreviated as BS, and there are 4 criteria like that, and then furthermore there are 7 criteria that are supporting a benign effect. What are this kind of criteria? I will not go discuss these criteria in detail, but there are some highlights that you can take in mind. So there are population data, and when the minor allele frequency is too high for a specific disorder, or the observation in controls is very high and it's inconsistent with disease penetrance, you can use this as a criteria that the variant is probably benign. To be pathogenic, of course, then we are talking about variants that are not very frequent in the general population. So you can assume a Mendelian disorder with high penetrance, that this kind of variants should not be too frequently present in genome databases. Furthermore, there is computational and predictive data. As you see, if there are very user-friendly prediction programs that can be applied, however, the evidence that's related to that should not be taken into, should not weight too much in the decision tree. So that's why it's considered as rather in the supporting range. Pathogenic variants are really the loss of function variants that are occurring in BRCA1 and BRCA2. So these are is a very strong argument in favor of pathogenicity. There are also functional data that can be taken into account, but they only weight very strong if it's established, if it has been found as a pathogenic or as a neutral variant in very well established functional studies. And it's not always easy to know which literature you can rely on. Segregation data is not so easy to apply in the context of BRCA1 and 2 because these diseases are not fully penetrant. Also, de novo is more difficult to apply in the context of BRCA1 and 2 testing. A very useful criterion, however, is the allelic data. So when you find a variant in trans with another, with the variant, well, when you find a variant of unknown significance in trans with an with a variant, a true pathogenic, clearly pathogenic variant, this is a strong argument against pathogenicity. And then you can also use different databases, but we will come back to that later. Or other data like specific phenotypes are less applicable in the context of BRCA1 and 2 testing. Well, to be honest, I do still not know all these criteria by heart. So this is something you can use as a poster on your in your office to support you when you're doing the variant classification. But how many criteria do you need now to classify a variant as pathogenic or likely pathogenic or benign, likely benign, or when do you fall into the uncertain significance? So you can accumulate the different criteria, and then you can calculate if it falls into the pathogenic category. So for instance, when you have one very strong argument in combination, so you need, it's really an and that's written here, you need at least one other strong argument, or you need at least 2 moderate arguments, or you can have combination of 1 moderate and 1 supporting argument, or you can have more than 2 supporting arguments. If you do only have 2 strong arguments, then you can, if you have 2 strong arguments, it can also be classified as pathogenic. If you have only 1 strong argument, you need some moderate or additional moderate or supporting criteria, which you can read here. And in that case, we say the variant is pathogenic. Likely pathogenic, this means that you have less strong arguments. So you might have, if in case you have 1 very strong argument and 1 moderate argument, then you can classify it as likely pathogenic. A strong argument in combination with moderate argument, or a strong with 2 supporting arguments, or 3 moderate, or 2 moderate and some supporting, or 1 moderate and 4 supporting, leads to likely pathogenic. Again, this might also be a slide you can put on your wall when you're doing the classification, that you can go back to these criteria. And there are also some rules to classify a variant as benign. So you have 1 standalone argument, or you have 2 or more strong arguments, then you can be sure that the variant is benign. A likely benign variant is less severe, so you have 1 strong argument or 1 supporting argument, or 2 supporting arguments makes a variant likely benign. And if your variant does not meet any of the criteria listed above, you should say that the variant is falls into the category of uncertain significance. Also, when you have 2 criteria that are contradictory, then you should also put it in the uncertain significance category and not really use it for clinical purposes. So what's the process that you need to go through when you classify variants? Well, the first steps that you can take is just look up if you can find the variant in a database and if it has been classified there. So there are some variant databases, and you can also general variant databases like ClinVar, and there are also some locus specific databases like, for instance, LOVD, and especially for BRCA1 and 2. There are several specific databases that will be listed on the next slide. You can also spend a lot of time doing the literature search, because BRCA1 and 2 are, I think, almost the most sequenced genes in the world. So there are a lot of literature can be found about it, but it's really worth the effort to have a good classification, especially for missense variants. It can help you a lot. Segregation studies may be helpful if you do not have relatives available in your own lab, which will often be the case. You can also go to the literature and look up the pedigrees that have been published with your specific variant, and this can also help you in the classification. Also, functional studies, the outcomes are published in several nice papers. However, at the moment, there's not yet really consensus about the studies, well, about which functional study can be used as the gold standard. But as a general rule, if it has been investigated in several labs with different approaches and they have uniform outcome, you can use it. And then as a last parameter, you can check the in silico studies, so the predictions that are generated by the prediction programs. Here you see an overview of variant databases that can be applied. To quickly look up if a variant is, how frequent a variant is in a general population, you can use dbSNP and also GNOMAD, and the EXACT databases are very good sources to evaluate the allele frequencies in general population. In ClinVar, you can find listed in a more clinical context the evaluation of the variants, and some labs really put some nice information that helps you for the adequate interpretation. As locus-specific databases, we can advise the big database. However, this is not updated anymore, so you should be careful with the information that's available there. There is the Leiden Open Variation Database, so LOVD IRC, which is which contains a classification, but not always information on which this classification is based, but you can then contact the submitter. There is the BRCA Share initiative from the French labs, which was formerly called the UMD BRCA1, and this it also contains also contains interesting information. And then the BRCA Exchange has the ambition to combine the information of the different databases and can help you for the interpretation. So you will find there the information that can also be found in LOVD and in ClinVar. It's bundled there if the variant has been submitted to one of these databases. In what's concerning literature search, how should you tackle this? Well, Google Scholar is a good way of quickly checking if a variant has been published. You can use, you can just put BRCA1 C point and then the alteration you want to look up. But you have to be careful in the past. So before there were good agreements about numbering of nucleotides, the numbering may be different. So for the BRCA in the old big database, BRCA1 numbering for the nucleotides is 119 nucleotides more that were included, and for BRCA2 they started at position minus 228. So for the numbering at the moment today we have better agreements about that, and it's advised to use the LRG transcripts for variant numbering. Segregation studies has its limitations, of course, in the context of hereditary cancers, but it's especially useful if you see the occurrence of a BRCA1 or 2 variant with a known pathogenic variant on the other allele, and if the patient does not display Fanconi anemia phenotype, it's a strong argument against a pathogenic effect. And then the functional studies can also be looked up in the literature. Related to the in silico studies, so the in silico prediction programs, they evaluate evolutionary conservation. So they start from the idea that variants that have been conserved during evolution in different organisms, that they should be very important. The most frequently used are listed here. You have SIFT, polyphen2, align GBD, mutation taster. These are also the tools that are applied in the Alamut software, but they are also freely available through the internet. However, be careful, they are often contradictory, so they should not wait too much in the evaluation of your variant classification. And furthermore, we have the in silico studies that you can apply for splicing. Personally, I think these are more reliable than the ones to evaluate the missense variants. So there are 2 programs that have been validated by many authors and that have shown to be very indicative for which variants you can expect aberrant splicing. It's MAX and SCAN. So if you have, in case there is a more than 15% reduction to the natural splice site, for splice site find more than 5% reduction to the natural splice sites, this is an argument to further evaluate the variant at the cDNA level for classification, because then there is a chance, a quite big chance, that it will lead to aberrant splicing.
Thank you, Kathleen. My name is Dr. Emma Howard. I'm the Scientific Operational Director of the Northwest GLH. So first of all, I'm going to start to look at the expert classification for the BRCA variants that were included in Run 4. The BRCA variants in the context of ovarian and prostate cancer testing to classify to inform tumour response to PARP inhibitors are listed on this slide. Under ovarian cancer, there are 10 BRCA1 and 2 variants, and for prostate cancer, 5 BRCA2 variants were interpreted. So looking now in more detail and going to proceed to more worked examples are highlighted in red and include a more detailed presentation of this BRCA variant module for Run 4. So these are the different variant types that we interpreted on this module. The first one being a large-scale rearrangement in BRCA1. This was traditionally named exon 13, but is now named exon 12. It's LRG292T1, and this is a duplication. The exon start and stop coordinates of build 37 are on chromosome 17 at 4123 4421 to 4123 4592. The next one is a nonsense variant in BRCA2 at C.997680T, P.lysine 3326 Utr. The next two are in BRCA2, and they are frame shift variants at C.1013 del, P.alanine 338 frame shift Utr 11. The next one, also in BRCA2, is C.10095 delins GAATT AT serine 3366 asparagine frame shift termination 4. The next one is a 5' UTR variant in BRCA1 at C-107A to T. And the next slide lists all the missense variants, the first one being a missense synonymous variant in BRCA2 at C.570 G2A, valine 250 valine, and then a list of non-synonymous variants which are all missense, the first one being in BRCA1 at C.1508 A to G, lysine 503 arginine, and then the next one also a BRCA1 C.1648 A to C, asparagine 550 histidine. Next is also BRCA1 C.203 T2A isoleucine 68 lysine. Next one is in BRCA2 C.3620 A to G, tyrosine 1207 cysteine. Next one is a BRCA1 C.5071 A to G, P. threonine 1691 alanine. Next one is BRCA2 C.8351 G to A, P. arginine 2784 glutamine. And finally in that list, another BRCA2 variant C.7978 T to G, tyrosine 2660 asparagine. There's two splicing variants were also interpreted. These are both in BRCA2. First one C.316 plus 5 G2A, and the second one BRCA2 C.632 minus 3 C to G. So taking each example in turn, the first one was a rearrangement, and this is in BRCA1. cDNA nomenclature is described in the box at C.4185 plus 1, 4186 minus 1, to 4484 plus 1, underscore 4485 minus 1, and this is a duplication. The variant type was a large scale rearrangement, taking the next stage by looking into the databases, firstly looking at HDMD. This is the BRCA1 exon 12 duplication, and there are 3 separate citations, and HDMD classify this as a disease-causing mutation. Next is looking at the BRCA share database of the same variant. You can see that in the list of BRCA1 and BRCA2 in the columns, there is no co-occurrence with pathogenic BRCA1 variants, and BRCA share classified this as class 5 causal. ClinVar also demonstrated that there was multiple pathogenic submissions, and these are listed in the column on the right-hand side of the slide. So in summary, looking at this exon 12 duplication, the expert panel applied these list of evidence favouring pathogenic variant. The first one being PSV1 strong, because this is a duplication, it's presumed in tandem, and the reading frame is predicted to be disrupted by nonsense mediated decay. It's also cited in this paper, and we applied the strong category because we had to make an assumption this was the same duplication as described in papers previously. We then also applied PM2 moderate due to it being absent from control populations on the NOMAD database. PS4 moderate was also applied because the prevalence of the variant in infected individuals is significantly increased compared to the prevalence in controls. And lastly, we applied PP1 strong due to co-segregation with disease in multiple affected family members in a gene definitely known to cause the disease. There was no evidence favouring that this was a benign variant, and therefore the overall classification by the expert panel was that it predicted class 5 pathogenic. Turning to the second example, this is a nonsense variant in BRCA2 at 9976A to T, P. lysine 3326 Utr. And looking first at the database BRCA Exchange, they used the Enigma criteria to classify this variant as benign neutral. Likewise, HDMD, there is a number of citations listed, and the overall classification from HDMD is that this is a disease-associated polymorphism with additional supporting functional evidence. Likewise, BRCA share was found to demonstrate that this variant has been seen to co-occur with two other pathogenic BRCA2 variants, and they classified this as 1 neutral. LOVD database also had 81 submissions submitted to it, and they classified this as a benign variant. ClinVar, as demonstrated on the right-hand side of the slide, showed multiple submissions, all of which were benign. And the Nomad frequency database showed that the allele frequency for this variant was 0.006468, and it was also shown to have 14 homozygotes. This is an example of the splice prediction taken from a screenshot in Alamet, and there was no significant differences demonstrated between the 4 splicing tools, so there was no effect on splicing. And likewise, this shows conservation among species, and the bar shows where the amino acid lies, and the region is not highly conserved. Therefore, in summary, there was no evidence favouring that this was a pathogenic variant. However, the evidence favouring that this was a benign variant and was applied in terms of BS1 strong, in that the allele frequency was shown to be greater than expected for the disorder at greater than 0.6%. The expert panel also applied BS2 strong, as it was observed in 14 healthy adults in a homozygous state, and full penetrance in homozygote form would be expected at an early age. The other evidence that was selected was that BRCA2 truncating variants downstream of the residue P.3309 in BRCA2 are not clinically important, and this was demonstrated in a paper by Mesman in 2018. Also, there was another paper by Higgs et al. That showed that there was no evidence for this variant associated with an increased cancer risk. And taking all this into account, the prediction by the expert panel was class 1 benign.
So thank you very much to Emma. My name's Miranda Durkee. I'm one of the assessors and I'm based in Sheffield in the UK, and I'm going to take you through another few worked examples. So we're going to start with a frameshift variant. This one is a single nucleotide deletion in the BRCA2 gene, C.1013del, and that is predicted to cause the protein change of a P.alanine 338, then causing a frame shift with a termination 11 amino acids downstream. So as this is a frame shift, this generally has a prior risk of pathogenicity being very high. This particular variant isn't in a known functional domain, but as it's located in exon 10 and causes an outer frame transcript, it will be subject to nonsense-mediated decay. So therefore, we can use the PVS1 decision tree, and as it's a null variant in a gene where the loss of function is a known mechanism of disease, we can use PVS1 at very strong level. In addition, it is absent from controls in the population database Nomad, so we can also use PM2. For evidence favour it being a benign variant, we didn't find any. In addition, this variant is not cited on BRCA, BRCA Exchange, ClinVar or HGMD. And using the Tartigian 2018 modification of the ACMG guidelines, we can now classify one very strong and one moderate as pathogenic. So this gives us a class 5 pathogenic classification. Okay, on to another frame shift example, this time in the BRCA2 gene, and this one is a del-ints. So it's a deletion of the single nucleotide C.10095 and then an insertion of 11 nucleotides. And the predicted protein change is P.serine 3366 to asparagine with a frame shift 4 amino acids downstream. Again, generally, the frame shifts have a prior risk of pathogenicity being very high. And again, it's not in a functional domain, but importantly, this one is located in the last exon of BRCA2, therefore will not be subject to nonsense-mediated decay. And so you have to be very cautious with these types of variants. So looking at the evidence for pathogenicity, well, the evidence favour it being a pathogenic variant. It is listed in this paper by Zhang et al. 2018, it's also on HGMD, as pathogenic in pancreatic cancer. But if you look at the paper, there's actually no supporting evidence supporting that classification. The evidence favouring it being a benign variant is it is actually present on NOMAD, but it's a bit of a tricky one because it's not listed with this HGVS. It's actually listed as 2 separate insertion events. But if you click on either of those events and then scroll down the page to where you can see the BAM file, you'll see that both those insertions are located on the same allele in cis. And if you combine those insertions together, it gives you the same HGVS as our variant. So it's actually found on 69 alleles on NOMAD, which is at 0.02%, so we can use the BS1 classifier. In addition, from the BRCA shared database, it was found to co-occur with another BRCA2 class 5 frame shift variant. Therefore, we can use BP2. It is also located on ClinVar 9 times as likely benign or benign and 3 times as a VUS, and is reported on LOVD 28 times as either VUS or benign. So this is all just indicating that it's fairly common in the population. In addition, the other evidence that is actually quite tricky to incorporate into the ACMG framework is that we know this is downstream of the common P.lys 3326 truncating variant that's just been presented by Emma. And we also know that from this functional study by Mesman et al. That the P.3309 truncating variant was shown to not be clinically important. So in summary, we can classify this variant as a class 2 likely benign. Okay, on to my next example. This is a very interesting variant located in the 5' UTR. It's in the BRCA1 gene, C.-107A to T. And generally, variants in the 5' UTR we think of as having a fairly low prior risk of pathogenicity, but obviously this region can contain the promoter elements, and there are other mechanisms of disease such as having an upstream open reading frame. So again, you still need to look through all the literature, etc., to find evidence. So in this case, the variant is on BRCA exchange but hasn't been reviewed, as is the case with many variants. But it is located on the LOVD database, and it actually gives quite a bit of information. So you can see it's been reported in 2 families, and both by Evans et al. 2018, and it actually gives you some further information about both these families. So family 1 is a 3-generation family with 3 affected females. It also says there's 4 non-affected carriers. Now, you really should not use the term carrier for an autosomal dominant disorder, particularly where there's no Medusa penetrance. So we should really say 4 non-affected heterozygotes, 1 of whom being female, 3 male. And in the second family, there were again a 3-generation family with 2 affected females and 2 unaffected heterozygotes, 1 female, 1 male, and they've classified it as pathogenic. So the evidence favouring it being a pathogenic variant, well, we can use the segregation data by counting up the informative meioses from the Evans et al. Paper in those 2 families. And we've done that according to the thresholds and the methodology set out in Jarvik and Browning. And so using this model, then you would disregard any non-affected heterozygotes where there's no reduced penetrance. So we end up with a segregation score of 1 in 16 for these 2 families, which would give us a PP1 strong. In addition, that paper also used multiple functional assays to show that the variant was associated with somatic epigenetic silencing caused by promoter hypermethylation. So they did both pyrosequencing and bisulfite PCR to show that there was 50% methylation at the promoter, and they also used RNA sequencing to show that there was loss of expression from that allele, and they did that in multiple somatic tissues. So while these functional variants don't fit neatly into the ClinGen SVI guidelines for functional variants by Brinnick et al., we felt that there was sufficient evidence to use this, and we'll use PS3 at a moderate level. So in addition, we can use PS4 moderate as it is observed in 2 or more unrelated individuals, and in addition it was not observed on population databases, so we can use PM2. There was no evidence favouring it being a benign variant, and it was not cited on BRCA, HGMD or ClinVar. So the final classification for this case is a class 5 pathogenic variant. Okay, onto my final variant that I'm going to work through. This time it's a synonymous variant in the BRCA1 gene, C.750 G-to-A, and that is predicted to cause no protein substitution at the valine 250. So generally the prior risk of pathogenicity for synonymous variants is low unless it affects splicing. So the evidence favour it being a pathogenic variant in this case is none. The evidence favour it being a benign variant is that there is no impact on the splicing prediction programs, and we've used the 4 tools located in Alamet. And in addition, this is a silent variant at a very weakly conserved nucleotide, so we can also use the BP7 classifier. So in terms of other evidence, there was a publication, Jarhel et al. 2017, that did both splicing and a transactivation study and classified this variant as likely benign. However, there were insufficient known pathogenic and known benign controls used for us to use the Brinnick et al. Framework, so we haven't used BS3 in this case. So in addition, it was on NOMAD in 11 alleles, so 0.008% of populations. It is on BRCA exchange as likely benign, LOVD as a VUS times 4, and on ClinVar as likely benign, and also a VUS on BRCA share, and was not on HGMD. And so our final classification for this variant is class 2, likely benign. So thank you, and I'm now going to hand over to my colleague Ian.
Thank you very much, Miranda. So I'll be taking on the next set of variants from this run of the BRCA variants. My name is Ian Berry. I'm the laboratory manager and NGS lead for the Yorkshire and North East Genomics Laboratory Hub based in Leeds. The next variant we're discussing today is BRCA1 C.1508 A to G, and at protein level lysine 503 to arginine. So this is a Missense variant, which is non-synonymous, and the prior risk of pathogenicity in the Parsons et al. 2019 paper was calculated to be low. So in terms of the ACMG evidence favouring both pathogenic and benign classifications for this variant, there are no criteria that can be applied. So the only evidence we could find either for or against pathogenicity in this variant was the Parsons et al. 2019 paper, which classifies it as having a low posterior probability of pathogenicity based on the observation of a single case in a UK lab in which this variant was inherited with a BRCA1 nonsense variant. But in this case, we're unable to apply the BP2 criterion as this is only a single observation. The variant is on NOMAD, which is an extremely low population frequency across all populations. It's also on BRCA exchange, ClinVar and ARUP, but not on HGMD or BRCA share. So the final classification for this variant, bearing in mind the lack of evidence for or against pathogenicity, is class 3, variant of uncertain clinical significance. So moving on, the next variant is also in BRCA1, C.1648A to C at protein level. This is an asparagine 552 histidine change. The gene here, obviously BRCA1, LRG292T1. The cDNA nomenclature is C.1648A to C, and this variant type is a missense variant with a prior risk of pathogenicity, which is unknown. So in summary, the evidence favouring pathogenicity in this case fulfill none of the ACMG criteria, but there's significant evidence favouring a benign interpretation for this variant. So the variant has an allele frequency greater than the maximum credible population allele frequency for BRCA1, calculated using the CardioDB allele frequency app. And this is again taken from the NOMAD frequency. So the BS1 criterion is applicable. The BP2 criterion is also applicable based on the fact that the variant has been identified in trans with a known BRCA1 pathogenic variant in the paper cited here, Judkins et al. 2005. Functional studies of the variant in using several different methodologies showed no deficiencies in BRCA1 function from the Giudigli paper in 2011 and the Caligo paper in 2009, allowing us to apply the BS3 criterion. And there are multiple lines of computational evidence that support benign interpretation in this case, allowing us to apply BP4. As stated at the bottom of the slide here, the variant is on the NOMAD database with a frequency of 0.026% across all population frequencies, and it's also on BRCA exchange, ClinVar, HGMD, and BRCA share. So in summary, the multiple benign strong criterion allow us to apply a prediction of class 1 benign for this variant. So moving on, the next variant is also a missense variant in the BRCA1 gene, C.203T to A, protein isoleucine 68 to lysine. As I say, this is a missense variant, and the prior risk of pathogenicity for this variant is uncertain. It's in the functional ring domain of BRCA1. So the evidence favouring pathogenicity in this case, from the functional studies of programmed functional studies performed by Finlay et al. In 2018 and Starita et al. 2016, suggests that this variant has a damaging effect on the function of BRCA1, allowing us to fulfill the PS3 criterion at strong level. The variant is absent from population control databases such as NOMAD, allowing us to apply the PM2 criterion. And the variant has been identified in multiple unrelated individuals with breast and BRCA associated cancers, allowing us to apply the PS4 moderate criterion. There are also multiple lines of computational evidence suggesting an impact on protein function, allowing us to apply the PP3 criterion. And with no contrary evidence favouring a benign interpretation, the summary of this variant is that with 2 moderate, 1 strong and 1 supporting criteria, we're able to apply a final classification of class 4 or likely pathogenic for this variant. The next variant is a missense variant, this time in the BRCA2 gene. This variant is C.3620A to G. And at protein level, it's tyrosine 1207 to cysteine. This is again a missense variant, and the prior risk of pathogenicity is unknown. So the evidence favouring a pathogenic conclusion for this variant, there were no ACMG criteria fulfilled. And the evidence favouring a benign interpretation of the variant, there is one piece of evidence here, BP4, whereby multiple lines of computational evidence indicate that there's no impact on splicing or protein function for the gene product. We also found the paper Dinez et al. From 2020, which cite this particular BRCA2 domain as a cold spot for missense pathogenic variants, but this is insufficient to reach the BP1 criterion according to the ACMG-AMP guidelines, so we haven't applied this criterion here. The variant is present on NOMAD at very, very low allele frequency of 0.0004%. It's also present on ClinVar, BRCA exchange, but not reviewed, and it's not on HGMD, LOVD, or BRCA share. So in this case, there are insufficient evidence criterion to move this variant from a class 3 uncertain significance conclusion. Moving on to yet another missense variant, BRCA1 C.5071A to G, at protein level 3-anine 1691 to alanine. This is a missense variant in the functional BRCT domain of BRCA1 with an unknown prior risk of pathogenicity. The variant is present on BRCA exchange, but without, as of yet, a review. It's present on HGMD professional, and it's also on BRCA share, with currently classified as an unclassified variant or a VUS, but with one co-occurrence with a BRCA2 classified variant in a reported patient. The variant is also on the LOVD database, the classification of VUS. And it's on the ClinVar database with 6 submitters, all classifying as uncertain clinical significance. The variant has a C55 prediction from Align GVGD, which suggests it tends towards a pathogenic prediction. It's predicted to affect protein function using the SIFT algorithm and is considered to be disease-causing by the mutation taster algorithm. It's also possibly damaging using the polyphen-2 algorithm. There are very minor changes in splice site prediction, but not sufficient to suggest that this is an in silico prediction of splicing effect using some of the common splicing tools you see here. The variant also lies in the highly conserved functional BRCT domain of BRCA1. So in terms of the evidence favouring pathogenicity in this case, there are multiple lines of computational evidence supporting a deleterious effect on protein function, allowing us to apply the PP3 criterion. The variant is absent from large population control cohorts, such as NOMAD, allowing us to apply the PM2 criterion. And the variant has been identified in multiple unrelated affected individuals with BRCA-associated cancers, allowing us to apply the PS4 moderate criterion. There are no evidence favouring a benign prediction for this variant. However, the functional data for this evidence for this variant are somewhat conflicting. The Finlay et al. Programmed mutational paper indicates that this variant has an intermediate effect and therefore may be a hypermorphic variant on the genome editing assay, and a cisplatin assay, an olaparib assay, and a homologous repair assay performed by Bowman et al. On their paper in 2020 gave a neutral result, an intermediate result, and a deleterious result, respectively, leading us to a conflicting series of evidence that it's certainly not very declarative in either direction in terms of pathogenicity or benign for this variant. The variant's not on NOMAD, it's cited on BRCA share, HGMD, LOVD, ClinVar, and BRCA exchange, but not classified. So the criterion that we've applied here using the ACMG guidelines are not sufficient to raise this variant to a class 4 status, so it remains a class 3 variant of uncertain clinical significance. And that completes the variants that I will be talking you through today. Thank you very much.
Thank you, Ian. My name is Arjen Mensenkamp. I am a clinical laboratory geneticist in Nijmegen in the Netherlands, and I will talk you through the next slides. The first variant I will discuss is this variant in BRCA2 C8351G2A, which encodes a change of arginine to a glutamine. And this variant is in the DNA binding domain and has been tested by in several functional studies. And these functional studies show that this variant shows an impaired protein function. And besides that, there are multiple lines of computational evidence that will support a deleterious effect. However, recent data on multifactorial likelihood analyses, as published by Parsons, shows that this variant does not really segregate with disease, and also that there are some evidence for co-occurrence with other BRCA2 variants. Therefore, we have a conflicting data, and the final classification is class 3 at this very moment. I know there are at this moment studies going on to further reclassify this variant, but for now this variant is still a VUS. The next variant is also in BRCA2. It's the C7978T2G. This is also a missense variant within the DNA binding domain. And here we have multiple levels of evidence for pathogenicity. First of all, several functional studies have been done, and these studies have been given a strong evidence level for PS3, as published by Burnage et al. Last year. Also in the same papers, segregation was shown in multiple families and with over 10 meioses, which also within the ACMG criteria correlates to a PP1 with a strong modification. Besides that, the variant has been observed in multiple unrelated affected individuals in several families, and it is absent from controls in the NOMAD population database. And in addition, there are multiple lines of computational evidence which support a deleterious effect. Taken together, this variant is classified according to ACMG as a pathogenic variant. The next sample will be presented in more detail. It's a variant at the +5 position after exon 3, so in the interim 3 at 316 +5 G2A. This variant is well studied and is described also on the BRCA exchange page. And the BRCA exchange page will also refer to the Enigma classification in this case, which summarizes all the work that has been done on exon 3 skipping within BRCA2. This variant has been tested extensively, and this variant leads to an almost complete skipping of exon 3. And both this variant, but also loss of exon 3, which is in frame, is pathogenic, and therefore this variant is classified as pathogenic according to the Enigma classification. This is also reflected in HTMD. In HTMD, you will find all literature references to the variants, and exactly those papers that were also used in the classification here are summarized, that it will lead to a skipping of exon 3 and also occurred in several populations in breast and ovarian cancer. So HTMG classifies as a disease causing, but you have to be very careful with the HTMD classification, as this is not on a diagnostic level of classification. Within the BRCA share, which is what formerly was the UMD, the French UMD BRCA2 mutation database, summarizes all the information that is available for this variant. Already, like I said, the Enigma classification is presented here. It's not present in population databases, and also all the evidence that is available from ClinVar is summarized in here. And the BRCA share database classifies this variant as a pathogenic causal variant. The LOVD database is an open platform database in which several labs can submit their variants, and it also extracts the information from the ClinVar database, and therefore variants will occur in both databases with the same records. It should be, at least. And this summarizes the results from several labs in which the variant has been classified from VUS to pathogenic. And like I said, the most well-represented database is the ClinVar database from the ClinGen consortium, in which mostly the American diagnostic labs submit their results, but also other labs. Especially the American labs also give the evidence within the ACMG criteria within the supporting information. The supporting information is again here pointing to a skipping of exon 3, which is pathogenic by itself, and the variant is thereby also pathogenic. And this is also seen in the different splicing prediction programs as here available through Alamut. And the predictions here are more subtle, except for Gene Splicer, which is completely lost for the splice donor site, while other prediction programs support a moderate decrease in strength. But there is, of course, now functional evidence that this variant indeed leads to a almost complete loss of exon 3. In summary, we have a PS3 strong evidence for functional studies, where 2 studies show that exon 3 is lost from the transcript. And in addition, there is a PP1 upgraded to strong evidence due to the massive segregation data that is available for this variant. And if also taken into account that this variant is absent from controls, together within the ACMG criteria, this will lead to a prediction that this variant is pathogenic. And another splice variant in the acceptor splice site, here is in BRCA2, C632-3C2G. This variant was classified based on the multifactorial likelihood analysis by Parsons et al. As being pathogenic. In favor of pathogenicity is the functional studies that has been done for this variant, and that is a full splice site mutation on the strong level of PS3. In addition, this variant was observed in multiple unrelated affected individuals. So based on the ACMG criteria, based on these 2 items, this variant is likely pathogenic. So that is the slightly lower level of evidence than based on the Enigma classification, which most people will use. And although the difference is quite small in here, we hope that in the future the ACMG criteria and the criteria used by Enigma will be further merged. And we expect that within the beginning of 2021, there will be ACMG criteria adapted specifically to BRCA1 and BRCA2 in the near future. In summary, these are all the variants that we have discussed in this webinar today. Like I said, all the classifications were done based on the ACMG criteria. And there are several other criteria. Several other criteria are being used by labs. And we hope that in the near future, there will be a more unified classification criteria for BRCA1 and BRCA2 on the ClinGen platform in 2021. Thank you.